After studying this module, you shall be able to know iron-sulfur proteins, categorize iron-sulfur proteins based on the number of iron and inorganic sulfur atoms in the cluster, analyze the structure of iron-sulfur clusters, learn the characteristic features of iron-sulfur protein, identify the biological role of iron-sulfur proteins, know the synthetic models of iron-sulfur protein centers. Introduction. Proteins containing iron can be classified into three different groups: heme proteins, iron-sulfur proteins, and other iron-containing proteins. Iron-sulfur proteins, abbreviated as FeS proteins, are characterized by the presence of non-heme iron which is bound to sulfur from the cysteine residues in the polypeptide backbone and or to the inorganic sulfurs. Iron-sulfur clusters are prosthetic groups commonly found in variety of metalloproteins that can participate in oxidation reduction reactions and catalysis, such as ferredoxins, NADH dehydrogenase, hydrogenases, coenzyme Q cytochrome C reductase, succinate coenzyme Q reductase, and nitrogenase. Further, these iron-sulfur proteins fall into two major categories. One is simple iron-sulfur proteins and the other is complex iron-sulfur proteins. Simple iron-sulfur proteins consist of either one or more iron-sulfur cluster, whereas the complex proteins bear additional active redox center as flavin, molybdenum or heme. EPR spectra are often used for the detection and identification consist of iron-containing proteins. Has been the area of bioinorganic chemistry, which has modernized over the decades. Since the group of non-heme iron-sulfur clusters are involved in electron transfer reactions within the biological system, they therefore are ubiquitous in nature. They are thus thought to be of fundamental for the processes like photosynthesis, nitrogen fixation, and various metabolic reactions. But still, the precise nature of their composition and stereochemistry was not established until very recently. In fact, it was only during the period of 1952 to 63 that these iron-sulfur compounds were first recognized as a consequence of discoveries made in the field of anaerobic fermentative metabolism and photosynthesis. The main reason behind this undiscovered important class of compound was that the iron-sulfur redox centers which were responsible to form the active sites of these proteins could not be extracted from the proteins. All the attempts to extract iron-sulfur proteins caused the disintegration of the active site of the protein, which further resulted in the complete loss of biological activity from iron-sulfur proteins. Finally, the method which became suitable for their detection was low temperature electron paramagnetic resonance spectroscopy, and during 1968 to 71, X-ray crystallographic results provided the exact composition and the stereochemistry of iron-sulfur protein active sites. Biological role of iron-sulfur proteins. The chief role of the iron-sulfur cluster is to facilitate electron transfer. Besides transferring electrons, iron-sulfur are best known for their role in photosynthesis, cell respiration, nitrogen fixation, in the metabolism of H2, NO2- and SO3 2-, oxidation and reduction. Also, iron-sulfur clusters have been recognized as sites for non-redox and redox catalysis. Examples include the exclusively iron-sulfur dependent hydrogenases and nitrogenases, as well as dehydrogenase isomerase enzyme of the aconitase type. Both the complex 1 and complex 2 of the oxidative phosphorylation have multiple iron-sulfur cluster. They are also responsible for the generation of radicals as illustrated by SAM-dependent enzymes and as sulfur donors in the biosynthesis of lipoic acid and biotin. Additionally, some iron-sulfur proteins regulate gene expression. Iron-sulfur proteins are vulnerable to attack by biogenic nitric oxide. It also serves as iron sensors. Now coming to the classification of iron-sulfur proteins, the characteristic features of iron-sulfur proteins are the iron atoms at the center of the tetrahedron ligated to the terminal thiolate sulfur centers which are coming from the cysteine residues or from the labile sulfur groups, either 2 or 3. 3 distinct kind of iron sulfur proteins with these features are most common. The first one is Rubredoxin, iron 0 sulfur, abbreviated as RD. This protein belongs to bacterial origin and was first isolated from Clostridium pasturanium, but was later found in the number of anaerobic microorganisms. They comprise of one iron atom which is tetrahedrally coordinated to 4 sulfur atoms which are coming from the cysteine residues present in the protein chain of 55 amino acids. This has been represented in figure. The redox center is composed of high spin iron 3 without inorganic acid labile sulfur in the core group. More than one iron center of this kind might be present in a molecule. Figure presents Rubredoxin. The Rubredoxin is a 1 electron transfer agent with Fe2+ and Fe3+ having high spin configuration. This is the equation. Ferredoxins, abbreviated as FD. Ferredoxins are acidic, low molecular weight and soluble iron sulfur proteins that are found in various organisms and act as multifunctional electron carriers in diverse redox systems. They comprise of those iron sulfur proteins which contain one or more clusters of 2 or 4 iron and labile sulfur atoms. They exclusively display electron carrier activity and not the usual enzyme function. Therefore, although hydrogenases, iron sulfur proteins containing one or more iron sulfur clusters, are enzymes, but they are not classified as ferredoxins. The existence of iron sulfur clusters of novel and unusual stoichiometry has now been proven with the help of chemical analysis, EXAFS and X-ray diffraction methods. 2 iron ferredoxins, represented as 2Fe2S. In ferredoxins, we have 2 iron ferredoxin, 3 iron ferredoxin, 4 iron ferredoxin and 8 iron ferredoxin. 2 iron ferredoxin is the simplest polymetallic system and a number of these ferredoxins have been isolated from plant, example spinach, and non-plant sources. Unlike Rubredoxin, the redox center of this type consists of a cluster of equal number of iron and labile sulfur atoms and is represented as 2Fe2S. This redox center is wrapped around the protein chain where the iron atom is coordinated to cysteine sulfurs. The cluster of 2 iron 2 sulfur protein can be considered as a bitetrahedral where the iron atoms are bridged by the 2 sulfide groups and additionally bound to 2 sulfur atoms of the cysteine group per iron atom. The oxidized proteins bearing 2 high spin Fe3+ ions are diamagnetic at very low temperatures, while the reduced form is paramagnetic corresponding to 1 electron. On reduction, 1 electron is accepted, formally giving 1 Fe3 group with the spin equal to 5/2 and 1 Fe2 group with the spin equal to 2, which interact giving a value for the dimer of S equal to 1/2, that is spin equal to half. In other words, 1 electron has been added to the diamagnetic system. This has been represented in the equation. Since the iron centers exhibit tetrahedral geometry, the crystal field splitting is therefore weak and both the metals are in high spin state in the oxidized form, whereas the reduced form contains 1 Fe3+ and 1 Fe2+ ions. Thus, these species exist in 2 oxidation states with 2 Fe3 and 1 Fe3 and 1 Fe2. One example of this kind is the chloroplast ferredoxin, which is essential in the process called photosynthesis. Coming to 3 iron ferredoxins, which is represented as 3Fe4S. Proteins are also known to contain Fe3S4 centers, featuring 1 iron less than the more common Fe4S4 cores, which is found in Bacteria digigas. Initially, from the X-ray crystal structure of the 7 iron ferredoxin from Azobacter wienandii, it was interpreted that this type of ferredoxin have 4 iron 4 sulfur and 3 iron 3 sulfur cluster. However, an error was detected in the analysis of this structure and it was corrected to have a non-planar Fe3S4 geometry. This structure seemed to be derived from 4 iron 4 sulfur core by the removal of one iron sulfur unit from one of the 8 corners of the cube. This cluster composition was supported from EXAFS data on the A1 landi ferredoxin. 3 sulfide ions bridge 2 iron ions each, while the 4th sulfide bridges 3 iron ions. Their formal oxidation states may vary from Fe3S4 positive, where all the iron ions are in +3 form, to Fe3S4 2 negative, where all the iron ions are in +2 form. In a number of iron sulfur proteins, the Fe4S4 clusters can be reversibly converted by the oxidation and loss of one iron ion to form Fe3S4 cluster. Example includes the inactive form of aconitase, which has an Fe3S4 and gets activated by the addition of Fe2+ and reductant. In oxidized form, all the 3 iron atoms are as Fe3+, and in reduced state, it contains 2 Fe3+ and 1 Fe2+. Next ferredoxin we have is 4 iron ferredoxin, which is represented as 4 iron 4 sulfur. This is the most widespread form of iron sulfur cluster in biology and has both plant and bacterial origins. The cluster contains 4 iron 4 sulfur as a redox center, and the iron and the sulfur atoms are placed at the vertices of cubane type structure, with each iron coordinated by cystenyl sulfur. This has been represented in figure. The result is tetrahedral of 4 iron and 4 sulfide atoms, with mean iron and sulfur distances being 2.75 Armstrong and 3.55 Armstrong respectively. Depicts 4 iron ferredoxin. Now this Fe4S4 electron transfer proteins may be further subdivided into low potential bacterial type and high potential HIPIP ferredoxins. Low and high potential ferredoxins are related by the redox scheme given in figures. In HIPIP, the cluster shuttles between 2Fe3+, 2Fe2+, and 3Fe3+, 1 Fe2+. The potential for this redox couple comes in the range of 0.4 to 0.1 volt. Two pair of oxidation state, Fe3+, 3Fe2+, and 2Fe3+, 2Fe2+, are found in the bacterial ferredoxins with the potential in the range of -0.3 to -0.7 volts. The two redox couples of 4 iron and 4 sulfur cluster share the Fe4S4 2 positive oxidation state, and the difference is attributed to the degree of hydrogen bonding, which strongly modifies the basicity of the cystenyl thiolate ligands. One more redox couple, which is even more reducing than the bacterial ferredoxin, is implicated in the nitrogenase. Besides this, some 4 iron 4 sulfur clusters attach substrates and are thus classified as enzyme cofactors. In aconitase, the iron sulfur cluster binds aconitite at one iron center that lacks a thiolate ligand. The cluster does not undergo redox but serves as a Lewis acid catalyst to convert aconitase to isocitrate. In radical SAM enzymes, the cluster binds and reduces S-adenosyl methionine to generate a radical, which is required in many biosyntheses. Another ferredoxin we have is 8 iron ferredoxin, which is represented as 8Fe8S. It is of bacterial origin, Peptococcus aerogenes, and consists of 2 Fe4S4 cubes, which are separated from each other by the distance of 12 Armstrong. We have other iron sulfur proteins as well. All simple iron sulfur proteins that neither fall in Rubredoxin nor ferredoxin belongs to the category of other iron sulfur proteins. It should be noted that there exist some iron sulfur proteins that have no redox role. Examples include endonuclease 3 and aconitase. Moreover, some complex polymetallic systems are known. Examples include both the 8 iron and the 7 iron clusters in nitrogenase, which is represented in the figures. Besides this, carbon monoxide dehydrogenase and the iron-iron hydrogenase also feature unusual iron-sulfur clusters, and a special 6 cysteine coordinated to Fe4S3 cluster was also found in oxygen-tolerant membrane-bound nickel-iron hydrogenases. Now coming to the synthetic models of iron-sulfur protein centers. In addition to understand the structure and the function of iron-sulfur clusters, a major component of the work has been the fabrication and the consequent study of their synthetic models. Synthesis of 4-iron ferredoxin center was first achieved by Holm and his group when methanoic solution of iron 3 chloride and sodium hydrogen sulfide was treated with 6 equivalent of NaSCH2PH. This resulted in an intensely colored solution of Fe4SiSCH2PH4 cluster. In this reaction, some of the thiolate ligand is consumed to reduce half of the iron formally to the +2 oxidation level required for 4 iron 4 sulfur 2+ unit. With a slight modification to the synthetic route, instead of NaHS, Garner and co-workers used FeCl, FeCl2 together with elemental sulfur in the presence of thiolate. The thiolate serves as a reducing agent to yield the tetranuclear species in a self-assembly reaction. Just after the preparation of tetranuclear cluster, the discovery of synthetic routes to the analogues 2 iron 2 sulfur family began, and the characterization of mononuclear Rubredoxin modules subsequently followed. The basic difference between the natural and the synthetic model system is that iron center is ligated to simple alkyl or aryl thio groups rather than the cysteine residues of the protein polypeptide, which is found in natural iron-sulfur proteins. In summary, we have iron-sulfur proteins are non-heme proteins and are found in bacteria, algae, fungi, plants and mammals. They have low molecular weight, 6,000 to 20,000, and contain iron, cysteine sulfur coming from the amino acid cysteine in the protein chain, and inorganic sulfur, also known as non-cysteine sulfur atoms. Their main role is to transfer electrons in the processes like photosynthesis, nitrogen fixation, metabolism of H2, etc. The sulfur ligands are arranged tetrahedrally about the iron atoms, and the presence of inorganic sulfur is indicated through the release of hydrogen sulfide gas when these proteins are treated with a mineral acid. This is represented in the equation. The cluster of iron and inorganic sulfur atoms in the form FeNSN prime or NFeN'(s), where N and N prime are 10, 22, 33, 44 respectively, constitutes the core group in these proteins and are their redox centers. These redox centers are wrapped in a protein chain with the cysteine sulfur of the protein coordinating to the iron of the core group. These proteins may be categorized into 5 accepted classes of iron-sulfur centers according to the numbers of the iron and inorganic sulfur atoms in the cluster: Fe1S0, Fe2S2, Fe3S4, Fe4S4 and Fe8S8. The classification is described in the table, where the iron-sulfur number, if it is 10, conventional name is Rubredoxin, and the number of electrons involved in the redox reaction is 1. In case of 2 iron 2 sulfur, the conventional name is 2 iron ferredoxin, with the number of electrons involved in the redox reaction is again 1. Another iron sulfur protein contains 3 iron 4 sulfur, which is also named as 3 iron ferredoxin, and has 1 number of electron which is involved in the redox reaction. The last is iron sulfur 4 iron 4 sulfur, with the conventional name as 4 iron ferredoxin, and has 1 electron which is involved in the redox reaction.